Good morning, everyone. Welcome to this section. Welcome to another lecture in the series of immunology. And today we'll be taking again, it's a series lecture. So we'll be talking about the MSc part. We had already started with the lecture of MSc and we had done understanding the concept of MSc class 1. Now we'll be talking today regarding your MSc class 2 molecule and perhaps the most relevant part because the class 2 is used or it is playing a very crucial role in terms of activating the T helper cells or presenting the process antigen by antigen presenting cells. So in that scenario, the eliciting the response of B cells and activating the T helper cell in both the cases, MSc2 is crucial. So we had a basic understanding of class 1. So taking that understanding, we'll try to understand the complete structural aspect of this particular class 2 MSc molecule, how the structure is folding, what is the antigen binding cleft, how this MHC molecule, and another set of information that we'll be reading in this particular lecture or try to understand in this particular lecture is after understanding class 1 and class 2, how they are appearing on the membrane. What is the process by which this particular class of MHC molecules attached to the antigen peptide and from inside of the cell they are going outside. So that complete aspect we'll be studying in this particular lecture, and that aspect is called as antigen presentation. So by the end of this lecture, I expect that we will be winding up this particular understanding of the MSc molecule and the antigen presentation. So let's begin with the class 2 molecule, a similar kind of structure that we had read in the previous lectures. Right, when we are discussing regarding the class 1 MSc molecule. So this is again a similar scenario, but what is the difference? See, it's also a heterodimer. It's a heterodimer. Why? Because it is made up of alpha chain and beta chain. There are two polypeptide chains. So if two polypeptide chains are coming together, we are calling it as heterodimer. Now, in the class 1 molecule, when the comparison was there, the structure was very different. Let's see here. I can divide this particular molecule with this line, and I will find a mirror image. I will find a mirror image. This is called as symmetry. Now, this mirror image, although structurally is a mirror, but the sequence-wise is very different. The alpha chain consists of two domains, alpha 1 and alpha 2. This alpha 2 is again a immunoglobulin-like domain. Now, this particular molecular weight of the alpha chain is ranging from say 32 to 34 kilodaltons, and in the previous lecture, we had a very glimpse that if you are doing an experiment, if you are working as an active culture, then how exactly you will be deciphering with the base of the experiments the constituent MSc receptor. Now, if the question comes, suppose you are working in a lab, you are going for an independent research. Now, a problem came. You want to study what is the composition of this particular MSc molecule, whether it is made up, whether this particular receptor is a single protein or it is made up of multiple polypeptides, or what is the conclusion that you will harness. So how will you go about it? How will you go about? We established a very basic understanding and we'll go and understand it again in that particular scenario. So before going to that, please understand that it is a two polypeptide chain that are coming together to form class 2. Similar was the case with class 1. But here you will see that alpha chain is made up of two domains and beta chain is also made up of two domains, alpha 1, alpha 2, beta 1 and beta 2. Fine. Then they have a pretty lengthy cytoplasmic tail. Both the chains are having the lengthy cytoplasmic tail. In class 1, that was not the case. Only alpha chain was the one which was having a cytoplasmic tail. Now, the molecular weight ranges from 32 to 34, whereas the beta chain molecular weight ranges again in the same region, but it is somewhat lesser when we compare it to the alpha molecule. Then the number of disulfide bonds, again the number of disulfide bond is 3 when we are talking about the class 2 molecule. Now, these both the alpha chain and the beta chains, when we had discussed the genetic composition, the regions that are identifying, the HLA terminology that we discussed, human leukocyte antigen terminology. In that category, class 2 is always coded by 3 regions as we discussed last time, that it is coded by DP, Q and R, DP, Q and R. So both these chains, alpha and beta chains, are coded by these three regions. So class 2 molecule in human leukocyte antigen of, or I should say class 2 MSC molecule of humans are coded from this particular segment of the chromosome, which is lying on chromosome number 6. And in that region, the region is represented by DP, DQ, and DR. Always remember, it's one of the very favorite questions of competitive exams if you're going for immunology. What is the genetic region that is coding for class 2 MSC molecule? And your answer will be human leukocyte antigen, DP, DQ, DR. Fine. When we are talking about class 1, it was HLA-BC. There was no definition of DP, Q, and R. So please remember in that term. Now, if we look at this particular structure, let's look at this particular structure. How will you bifurcate this structure? See, I can put a line here and I can put a line here. This is a membrane and there is a segment of transmembrane domain. This is a transmembrane region which is penetrating the membrane. Now, membrane, you know that on the surface it is hydrophilic, inside it is hydrophobic, and then again on the cytosolic part it is hydrophilic. So we have two transmembrane regions in MSC class 2. Two transmembrane regions in MSC class 2, and we have two cytoplasmic domains also in the MSC class 2 molecule. Now, when we are talking about antigen binding, the process antigen binding, this is the antigen binding cleft that you are seeing. In class 1 molecule, there was no gap. See, there is a gap here that I am highlighting. In class 1 molecule, there was no gap. It was closed. As I told you in the last lecture, it was closed. It was closed and it is made up of alpha 1, 2, and 3 domain. So that was closed. Here it is open. Here it is open. So what is the difference between open and closed? See, let me erase the ink here and let us understand. If it is open, and my peptide is this long, suppose this is my peptide. So my peptide will be coming and it can penetrate in from this cleft to the down part and it can attach in this form. Whereas if I close this particular cleft, suppose I close this particular opening, then my peptide or the antigen which was of this length will not be able to go inside. It will be holded in this manner. So the binding of this lengthy peptide will not be tight. It will be loose binding. Fine. So that is the major difference between class 1 and class 2. That in class 1 the cleft is closed, so the peptides which are binding to the class 1 molecule are small antigenic peptides, small antigens. Antigens after being processed are converted into small fragments and then only they can associate with class 1. But when we are talking about class 2, the relative length of the peptide antigen is increased and it can bind to as great as 30 amino acid antigen, whereas when we are talking about in the small, it is the length is around 12 amino acid. So you see that is the difference. It can accommodate the larger region. It can accommodate the larger region. And then MSC class 2 molecule, binded antigen, is recognized by the T cell receptors. These are presented by the antigen presenting cells. So these certain aspects you should always remember in order to have a better understanding of antigen presentation, that what is class 1, what is class 2 molecule. Fine. Then see, this particular domain beta 2 is very specific for CD8 that we have discussed, cytotoxic T cell CD8. So let us give a just brief comparison first of all, that how exactly we had talked about that particular aspect. So this is your, if I say the representation of your MSC molecule, then this in this manner it was getting folded and you are seeing here, and then there was a beta globulin which was attached to this particular part, and this is extending to a tail. This is your membrane. Fine, and this was the antigenic cleft that we were talking about here. Whereas right now what you have understood is there is an antigenic cleft here, and you are looking at two separate chains that you are focusing on in terms of your understanding. So this is class 2 This is class 2. Let us extend this part, and this is class 1. This is a small cleft that we are talking about, and this cleft is relatively larger if we are talking about. Then the difference you can understand in terms of your cytoplasmic domain, the differences are pretty much clear. The length is pretty much clear. Then again, this is beta chain, this is alpha 1, alpha 2, alpha 3. So we have 3 domains in class 1, whereas here only 2 domains of alpha are present, and beta is present in the form of 2 domains. So that is the thing that you should always understand. Please, please understand this is alpha 1 and this is alpha 2. And that is how the differentiation that what we have discussed right now is arising. Now, coming back to our question that how will you in lab, how will you come to know about the structural part of MSC molecule? It's very simple. You have immune cells, you want to study class 2 molecule. So class 2 molecule is present only in APCs. So you need the population of either macrophage you are talking about, or B cells you are talking about, or DCs you are talking about. So the percentage of cells that are present, you can take out macrophages also, you can take out B cells also, or you can study on the basis of the cell lines. The B cell line you can use, or B cells lymphoma cell line you can use raw cell line for macrophages. So the idea is, in the previous lecture when we discussed that based on the surface marker and magnetic sorting, you are isolating these particular cells. So what you have done, you have a population of, let's say, these APCs. Now this is the cell. Suppose this is a big B cell that we are talking about. Let's say B cell, what we are talking about, and this B cell is activated. Now it will start producing the MSC molecule on the surface. When I say activated, that means it has been exposed to the antigen. For macrophages, if you want to activate the macrophages or the raw cell line, you can use LPS or lipopolysaccharide as antigen. Now, what you need, if you are going for isolating the membrane receptor, you need the membrane fraction. So what you will go for cell lysis, and after cell lysis you will isolate the membrane fraction by, you may isolate it by centrifugation, you may isolate with different methods. You need a membrane fraction, and from that you need the complete proteins that are present on the membrane. You can isolate the complete proteins that are present on that membrane fraction. Now, a cell membrane contains different proteins, but we only want to have MSC2 molecule. We only want to have MSC2 molecule. So this right now, this protein fraction contains multiple proteins. So what you can do next, you can go for affinity chromatography. Affinity chromatography, you have an antibody which is specific for MSC class 2. So what you do, you tag those antibodies on a column. Predefined columns are available. You can use those columns. So these antibodies are specific for MSC molecule. You add this protein mixture inside this column. Only MSC will be able to bind these antibodies, and remaining fraction will be eluted out. So once the reaction and complete elution is done, what you will be seeing, you will be seeing a column which contains antibody which are bound to the MSC molecule, So that scenario you will be having. So what you have done till now, from a cell, you have managed to separate the MSC molecule. Now your job is to isolate or purify this from this particular complex. So you can add a high ionic strength buffer, which will disrupt the interaction between the antibody and MSC molecule. And with this buffer, this bond will be broken, and you will get in a beaker, you will get in a tube, you will get the complete MSC fraction as protein molecule. You can estimate the protein. Go ahead, estimate the protein with Bradford assay. You can estimate the protein, and then next job is, once you have this particular fraction, which is only MSC2. So your main aim was to check how many polypeptide chains are present. So I have this fraction of MSC molecule now. I have this fraction of MSC class 2 molecule now. Okay, so what next is what I did the protein estimation. Let's go and perform SDS page. Let's go and perform SDS page. I made wells. Here I added the ladder. By ladder, I mean it's a predefined protein marker that I am adding. This protein marker will give me different bands, and I know the molecular weight of these bands. It will be, let's say, 100 kilodalton. This is again let's say 60, then I have 40, then I have 20 and 10. I'm just giving you an arbitrary ladder format, although we have multiple letters that are. So it's a standard that I'm talking about, or in common term, the comparison ladder that we'll be taking out. Then you can load this particular protein in the desired format. The volume, the amount should be equal. The protein content should be equal. And what you will see that there are two bands that you are getting right now. A band here, a thick band here, and in this manner. So you are getting now two bands. So if you are getting two bands, you can clearly narrate that we have two different polypeptide chains. Two different polypeptide chains that are constituting the MSC class 2 molecule. One chain's one chain's molecular mass is around, it's around 32 kilodalton, whereas the other around 28 kilodalton, whereas the other one is around 32 kilodalton. So this is how people came to know that MSC class 2 molecule or class 1 molecule is made up of two chains, and this is their molecular weight. Fine. Then you can take out this fraction from here. You can excise this fraction, the gel you can take out, and you can go for eluting the protein, and you will get a pure, pure beta 2, pure alpha 2 chain, alpha chain. You can take out this, you will get a pure beta chain, and you can go for further analysis of like protein sequencing. You can go for mass spectrometry for deciphering what is the exact molecular mass that we are talking about, what is the length of the amino acid that we are talking about. So you can do n number of analysis after this particular scenario. So this is how a single statement that we had made is done inside a lab, and this is how we come to a conclusion that yes, that much effort is required inside a lab to even give a single statement of any science or any, especially in this case of immunology. So after that, you will be coming to know that yes, it is made up of two chains, the beta chain and the alpha chain. There is a peptide binding clef, and the structural prediction, you can see that this is an alpha helix molecule, these are beta plated, the secondary structure information you will be getting. So this is the thing that you should clearly understand, right? Then let us go and start our comparison of class 1 and class 2. Now, first of all, you should be very clear in mind where inside our body this class 1 and class 2 molecules are present and what is the distribution that we are doing. Okay, so class 1, I told you it is present in all nucleated cells, and class 2 is present in APCs. So let us see what do you mean by that. In all nucleated cell, among all the cells of our body, what are the type of cells that are giving very high expression of MSC class 1 and what are the cells that have low expression? So all the immune cells which have nucleus, all those cells will be giving you relatively higher expression of MSC class 1 molecule. Like let's talk about class 1. Let's talk about class 1. So T cells have very high expression, B cells have very high expression, macrophages have high expression. Other APCs like Langerhans cells, pair patches, they all have very high expression of MSC class 1. Whereas if you talk about the epithelial cells, they have low expression of class 1. Then hepatocytes, kidney cells, brain cells, they all have other nuclear cells, they all have class 1 molecule representation. And if you're talking about the neutrophils, which are very important aspect of our granulocytes, lymphocytes, they also have a very high expression of MSC class 1. But crucial point, non-nucleated cell, that is where you're talking about RBCs, there is no presence of class 1. There is no presence of class 1. Then class 2, only APCs. Please remember, only APCs. So we are talking about B cell, very high expression, dendritic cell, again high expression, macrophages, relatively less expression compared to B cells. Epithelial cells of the thymus that we are talking about, those are also antigen presenting cells, and they have a very high expression of class 2 molecule. whereas the remaining other nucleated or non-nucleated cells, they don't have this particular potential to present the antigen. They are not playing any role in antigen presentation. That is the reason why they don't have class 2. Or I should rephrase myself, since they don't have the potential to express the class 2 molecule, they cannot work as antigen presenters in case of immune reactions. So, uh, why we are talking about this particular scenario? We should only talk about immune cells. Why I am talking about the other nucleated cells? It's very simple. There are many antigens that can go and internalize in the cell, endogenous antigens. Like viruses, they can infect any cell and they will go inside the cell. Like parasites, like mycobacterium, tuberculosis, bacteria, what we're talking about, those all are endogenous antigens. So that is the reason why we're talking about in this particular scenario. So your MSC molecule, see, it is a recognition molecule. It will recognize the antigen. It will recognize the antigen. But this MSC molecule does not have any kind of specificity for any specific antigen or any antibody or T cell receptor. So it does not play any kind of role in giving a specific clonal propagation or increasing the specificity of T cell or antibodies. No, its pure function is just to go for antigen recognition. That's it. And it can bind to variety of antigen. It can bind to variety of antigen. The only requirement is the antigen first should be processed, degraded to small molecules, and then only it will be presented. So the presentation always happens in the cleft that we had discussed. So that is, the cleft that we had discussed, the antigen binding cleft, the region which is far away from the membrane. This antigen binding region is actually, if you go to the genetic studies, it shows polymorphism. So the variation in the amino acid sequence in these alleles are very high when we are talking about MSC molecules. So different allelic forms are giving you different results. So let's talk about the MSC1 MSC molecule, a comparison kind of chart. So we already understood the composition in terms of polypeptide chain. We know the distribution now, that nucleated and APCs, the composition of antigen binding cleft, the alpha 1 and 2 domain in MSC1 and alpha 1 and beta 1 domain in MSC2. Yes, we had discussed this also. Then binding to the T cell co-receptor, the class 1 is alpha 3 region is specific for CD8 binding. That's what we discussed. And MSC class 2, if we talk about, then the beta 2 region. The beta 2 region is specifically for CD4 molecule, the T helper cells. Then they can accommodate around 8 to 12, actually I would say 8 to 12 amino acid residue, whereas this particular class 2 can accommodate around 30 residues easily. So the nomenclature, if you talk about in terms of technical nomenclature, we are calling it as human leukocyte antigen ABC, and this we are talking about a human leukocyte antigen DRQ and T or PQR, as what manner you want to remember. So please remember this particular comparison chart for your future references and to have a better understanding of the narrations that we talk about in terms of your immunological aspects. So let's go ahead and talk about how the cells are now communicating. After understanding the T cell receptor, after understanding the MSC molecule, let us establish a very basic understanding of how these cells are going from contact. So when we are talking about cell to cell contact, yes, it can happen via cell surface receptor. The two signals or the two receptors that are very crucial, the TCR complex that we had discussed, complex along with MSC molecule, and the other that was that was also important, that is required for secretion of interleukin 2 for clonal propagation, was CD28 binding to the B7 receptor, Now that is the thing that you should represent. This is your T cell. This is your antigen presenting cell that we have taken the dendritic cell here, example of dendritic cell. Now cell to cell contact is required for activation, yes, but these immune cells also communicate without coming in contact, and that communication is provided by the chemical messengers. Now these chemical messengers, now, or how they are communicating, this T cell is releasing a chemical messenger, Now those chemical messenger which are carrying the information are called as chemokines or cytokines. If you are talking about cytokines, because they are giving information from a cell to cell. Now it has released a certain cytokine which will be taken up by these antigen presenting cells, and then the changes will start happening, and they have secreted a certain information in terms of cytokine which will be taken up by this cell, and then again changes will start happen. So this cross communication between the immune cells in the body is mediated by soluble secretory molecules or secretory compounds called as cytokines. And cytokines are in different categories. We'll be studying the different interleukins, the TNF factors, the interferons, the chemokines that we'll talk about in the coming lectures. So after understanding the chemical mediator, you should also understand that what's the difference between exogenous and endogenous antigen. I'll not go in detail. It's very simple. Exogenous, where we are getting exposed to antigens that are coming from external environment. Endogenous, the antigens or the peptides that are produced inside our body. Now, I am talking about antigenic peptide. Now, what are antigenic peptide inside our body? See, we have a normal cell physiological mechanism, biochemical reaction. Any deviation from the standard path, any mutation that is there, or any aberrant protein that is there, or any kind of viral infected cell if it is present, or any kind of cancerous cell that is present, those all categories are cumulatively forming your endogenous antigen group. So that is what you should have a very clear-cut understanding. Then antigens are categorized into three different aspects. First is autoantigen, then alloantigen, and then heteroantigen. What is autoantigen? Autoantigen is self-antigen which is belonging to the host itself. Then we have alloantigen that is derived from external environment, other members of the species. You can say like alloantigen is mostly when we are talking about in terms of transplantation or blood transfusion, where we are talking about the same species, but we are talking about different individuals. So if an antigen is acquired from other individual, that is called as alloantigen. Like in blood transfusion, if you are transferring the blood, then the blood surface antigen we are talking about in terms of B antigen and the other glycoproteins, those all are called as alloantigen. Then heteroantigen, of course, this is the main category of exogenous antigen where the species barrier is crossed and we are talking about two different species. So that is the thing that you should have a very clear understanding between what are the changes that are there. So autoantigen belonging to us, alloantigen from other members of the species, and heteroantigen from different species. That is, in nutshell, a very simple narration and very simple narration that how things are happening in this particular zone, how things are happening in this particular zone. So we have a very basic understanding of these particular antigens. Then there is another category that people have defined, TD and TI antigens. See, everyone defines based on their convenience. TD is nothing technical. It's just T cell dependent antigen and T cell independent antigen. Now, what is T cell dependent antigen? Antigens that require the help of T cells to produce an immune response. Or I should rephrase, our body, when it requires the help of T cell to mount an immune response against the antigen, we call those antigens as T cell dependent. And if our body is mounting an immune response with the help of B cells following the humoral axis, then those antigens are called as T cell independent antigens. T cell independent antigens. So if my body is clearing a pathogen based on antibody only, then there is no involvement of T cell. I'll be calling it as T cell independent antigen. And if my body is making the memory T cells and it is going for cell mediated immune responses also, then I'll be calling it as T cell dependent antigen. And the classical example of T cell independent, T cell mediated humoral response is your lipopolysaccharide, LPS of the bacterial source, that is T cell independent antigen, because it is majorly cleared from macrophages and T cells. Then there is another terminology that you should be very clear, called as super antigen. Now, what is super antigen? There is an antigen. Then what is super antigen? See what super antigen does. Super antigen will be binding to the complex. What complex? MHC class 2, which is bounded to the T cell receptor. This is in general what we have read. Now the super antigen will be going and binding to this particular complex, and what it will do? It will generate the activation, super activation of this T cell. Initially, this T cell was supposed to get activated and give you the immune response. Secrete the cytokines 2, 4, 6, 5. Fine. But as soon as super antigen bind, this T cell became hyperactivated. Now the condition is that it is very much activated. It is feeling a sense of emergency, or there is certain sense of rapid functioning. So instead of producing, let's say, 10 molecules of IL-2, it produced 100 molecules of IL-2. So what is there? Your body is getting a storm of cytokine. So this condition is called as cytokine storm condition, because the immune cells are now hyperactivated, and those molecules that induce this hyperactivation, that induces these cytokine storms, are called as super antigens. And they always bind to this particular complex of T cell receptor, peptide, and MSC molecule, and they are specific for these regions. They both are specific. The super antigen, let's talk about. It contains two domains of binding. One domain can bind to T cell receptor, and another domain can bind to class 2 molecule. And they similarly they will go for increasing the cytokine storm. So what will happen if cytokine storm is there? What will happen if cytokine storm is there? See what happens. The T cells, exactly, the T cells which are supposed to get activated, will now be deleted because they have exerted out, and these all the T cells that are required to mount a response will now be deleted, and super antigen, because of the cytokine storms and deletion of the T cell, will result in immune deficiency. And that is how the functions are happening, and that is how the complete functioning is happening. Then we have another aspect of host defense, how our host is mounting the defense against. You can talk about the intracellular antigens or endogenous antigens, the virus infected T cells. The cells that has taken up the virus will process the virus, and it's actually after processing, it presents the virus antigen with the class 1 antigen, which will lead to the activation of cytotoxic T cell. Whereas if you talk about, let's say, Mycobacterium tuberculosis, which is again a classical example, again it is presented on the surface, which is recognized by the T helper response first, rather than going for cytotoxic T cell, and that can be easily visualized. So a virus infected cell always activates T cells, cytotoxic T cells, whereas when we are talking about the other endogenous antigens, then they are presented by the antigen presenting cells, and then they elicit the T helper cells response. So always remember that particular aspect. Now there is another very interesting finding, that when we are talking about the antigen will be binding to the MHC1 molecule, the antigen that will be binding to the MHC1 molecule, is there any kind of specificity or any kind of unique property that is required in those antigens or antigenic peptides? So people have analyzed these particular aspects also, and what they concluded, I am giving you a glimpse. See here, this is 1, 2, 3, and 4 different peptides that can bound to class 2. What is common? The common is the presence of tyrosine residue. Here again, tyrosine and phenylalanine residue. So the presence of aromatic amino acid. The presence of aromatic amino acid is responsible for the anchorage of peptide in the cleft. Anchorage of peptide in the cleft. And also, along with the aromatic amino acid, the hydrophobic amino acids like leucine, valine, isoleucine, these are also responsible for the anchorage. So when we are talking about the class 1 molecule, the peptides which are having aromatic amino acids and the hydrophobic stretch of hydrophobicity in the amino acids are actually anchoring that particular cleft using MSC class 1. Or when we're talking about MSC class 2, don't worry about these. You are not supposed to remember these sequences because these sequences can be from any peptide. But what you need to understand is the presence of D, that is aspartic acid and glutamic acid. So the acidic amino acid, and the presence of again the hydrophobic residues are required if an antigen has to bind to the cleft of class 2. So for class 2, we require the antigen should have a stretch of acidic amino acid and hydrophobic amino acid, whereas for class 1, it should be aromatic and hydrophobic amino acid. So this is what I wanted you to understand before moving ahead to your antigen presentation part, that you should have a basic understanding of what type of scenarios are we talking about, what type of MSC molecules are we talking about, what are different terminologies of antigens. And so right now we have established the structural aspect of all the MSC molecules. Now in the next part, we'll be focusing on the antigen presentation via MSC molecule. So these are certain MCQs that will again help you in better understanding, like which of the following is true for the antigenic peptide that is presented by class 1 and class 2. Which is true? They are non-covalently but stability bound. They are covalently bound to the MSC. The antigen peptide dissociates. And what is the purpose? So exactly if you are going about in understanding in terms of your MSC molecule, then you should have a better clear-cut understanding how these things are getting processed. Okay. So that is it. Thank you so much for your patience listening. We'll again meet in another series of the lecture. Till then, take care.